My name is Susan Shaney, and I'm the Sheep and Goat Specialist for University of Maryland Extension. Today, we're going to do our second webinar in our worm webinar series. The topic is fecal egg counting. These webinars are sponsored by the University of Maryland, Delaware State University, Virginia State University, and Fort Valley State University. Presenters include myself, Dr. Dahlia O'Brien, who will present today, Dr. Kwame Matthews from Delaware State University, and Dr. Nikki Whitley from Fort Valley State University. Today's topic is fecal egg counting primer. Again, our presenter is Dr. Dahlia O'Brien. Dr. O'Brien is the Small Ruminant Specialist at Virginia State University.
Okay. Hello, everyone, and thank you for joining us today. As Susan said, I'll be talking to you today about fecal egg counting, and we'll be going over why we do fecal egg counts and the steps in conducting a fecal egg count on your own if this is something that you're interested in. So first off, what are fecal egg counts? So fecal egg counts are simply they're a quantitative measure of the specific numbers of eggs present per gram of feces. This is done using a known quantity of feces and a known amount of flotation solution. A fecal egg count being a quantitative measure, this is in contrast to qualitative analysis that can also be conducted, which they basically show the presence or absence of eggs. So, and this is typically what's done maybe in a vet office to see if your animals have worms or not, where they simply mix the fecal matter in with a solution, fill it up in a tube, put a slide over it, and just look under the microscope to see if there are any eggs present or not. It doesn't give you a quantitative measure, so it doesn't tell you how many eggs per gram of feces are actually in there. And these are mostly done just to show general trends and to see, just as I said before, whether or not your animal might be suffering from parasites. So what exactly do fecal egg counts tell us? Fecal egg counts can help us to monitor the rate of pasture contamination. All right, so we could use those to essentially determine when to move animals off of a particular pasture. In doing this, you would need to collect random samples, and it will just give you an idea of just how wormy the animals are. Will be a direct correlation to how infested your pastures are. So for instance, if you have 40 animals and you want to get a good estimate of the group, you would take about 12 to 15 random fecal samples, and then you could do a fecal egg count. And if this is relatively high, this will indicate to you that maybe your pastures are heavily infested and it's time to move those animals onto a new pasture. So it could be used as a good tool to monitor pasture contamination. It's also a great tool to determine drug resistance. If you are unsure of how effective drugs are on your farm, or that current drug that you're using is, then you could do what's known as a fecal egg count reduction test. And this simply requires you doing a fecal egg count prior to treatment, treating the animals, and then taking a sample 10 to 14 days after. This typically requires, for the best and most accurate data, it typically requires a specific number of animals to test. You would usually want to test between 10 to 15 animals per dewormer, and you'd also want to have a similar size group as a control, just that this gives you a better idea of what's really happening to that infection in your herd. You would need a minimum fecal egg count to do fecal egg count reduction tests. So any animals included would have to have a minimum fecal egg count of 250 eggs per gram. And there are techniques out there for doing both individual or pooled samples to maybe cut down on some of the work that you'd have to do. What's nice about this fecal egg count reduction test is that it gives you a percentage resistance. Especially when compared to the larval drench rite assay. It tells you specifically what reduction was observed, and so you can have a better idea of how effective that drug is on your farm. Fecal egg counts can also help us in selection. It can help us to identify those animals that we want to include or keep back in our flock or herd, and also those that we might want to cull and get rid of. Parasite resistance and resilience, as you all know, they're both heritable traits. And essentially, an animal that's resistant will have certain traits. These animals, when you check them biweekly or how often you're doing FOMACHA scores, you will observe that these animals consistently have low FOMACHA scores. They rarely require deworming, and if you take a fecal sample, they also have low fecal egg counts as well. On the other hand, you have parasite resilient animals, and resilient animals, they will consistently have low FOMACHA scores when you're going through doing your checks. Have no other signs of parasitism. If you're basing deworming simply on, however, when you do fecal egg counts, then they have a high fecal egg count. Both of these animals are productive, but we do have disagreements from time to time about which animal is preferred. There's some people who prefer a resistant animal, and then there's some who prefer a resilient animal. And again, a resistant animal, basically they're able to limit infection, whereas a resilient animal, they can tolerate or withstand infection better. I tend to prefer a resistant animal. I want animals that limit infection, because resilient animals, even though they don't need deworming themselves, they can still contribute a lot of parasite eggs in their manure on pasture. And whereas this is not a problem for them, it could be a problem for susceptible animals in that group, or animals that you'll move onto that pasture. Conducting routine fecal egg counts will help you to identify those animals that are truly resistant and those that are resilient. So if you're doing your FOMACHA scores during your summer months and you're just looking to determine deworming, who to deworm or not, then that's fine. If you're looking at a group that you're thinking about selecting some animals out of to put in your herd or your flock, then I would recommend that you include routine fecal egg count and get at least 2 samples, and not only from the animal that you're thinking about collecting, but from a group of animals, because you want to you want to have something to compare that fecal egg count to. And then you could choose, if an animal consistently has low fecal egg counts, low FOMACHA scores, rarely requires deworming, that's a truly parasite resistant animal, and you can select that one and keep it in your herd. The question we get asked often is, should fecal egg counts be used to determine need to deworm? We get this question a lot, and we always get asked for a limit, a cutoff. What fecal egg count should I use to deworm my animals? If you look over on the top image on this slide, you'll see that there is actually a chart there. It comes out of Australia, where it actually gives the livestock species. It doesn't have goats on it, but it has sheep and cattle. It has the worm species. You can see the Haemonchus contortus, Teledorsasia, and Trichostrongylus. And it also gives you clinical significance. As you can see, Haemonchus contortus, or the barber pole worm, the clinical significance, you start seeing clinical signs when you have eggs per gram exceeding 2,000. That's when it becomes clinically significant. But we want to remind you that this will differ by animal. Again, we just talked about resistant and resilient animals. There's some animals that limit infection, and there's some animals that can withstand infection. So you might have some animals in a group that a couple thousand eggs per gram does nothing to them, whereas another animal that might be clinically significant. So we want you to think about the whole picture and not just use fecal egg counts as a standalone for deworming. And that's why you'll see I put on the slide, should fecal egg counts be used to determine need to treat? In most cases, no. Deworming is advisable only if an animal has a high fecal egg count, even if it has no other indicator of parasitism, because what this does, it limits your pasture contamination, and it limits exposure of more susceptible animals to those worm larvae. You should always use fecal egg counts in conjunction with FOMACHA and the 5-point check. And you can also use it along with weight gain or loss to determine need for treatment, but it should not be used just as a standalone. Okay, and remember too, you also need, at the bottom of that slide, you'll see that you also need to consider nutritional and physiological status and age of an animal. And this could be, for instance, think about a lactating female with multiple nursing young on her. If an animal like this that has a fecal egg count of a 2,000, you might decide to deworm her. Whereas if it was an open animal, just grazing with other adults out in the pasture, that animal, if she's showing no clinical signs, then that would be fine to not deworm her. All right, so today we're going to discuss the most common method or counting technique for ruminants, and it's known as the modified MatMaster technique. There are other methods that are out there, but this is the method that we use in our lab, and it's commonly used in small ruminant parasitology. It's based on the principle of differential density. So parasite eggs sink in water and they float in more dense solutions. So they really need a solution that's super saturated so that they can float to the top, and we're able to count them on the MatMaster slide. So we use this technique to count the number of oval-shaped strong-gile or trichostrong-gile eggs. And those are your barber pole and related worms. They're all shaped the same. They all look the same under the microscope, and you cannot differentiate them by species at the egg stage. It's very difficult to do that. In order to differentiate the worms and know what species they are, you'd actually have to take it a step further and do a larval culture on a fecal sample. In order to do the modified MatMaster technique, you will need a modified MatMaster slide. And these are available at the websites provided. You'll see that you have 2-chambered and 3-chambered slides, and you can get them at vetslides.com or examine.com. The 2-chamber slide, they generally run from about $17.95 to $20, depending on how many you purchase. The 3-chamber slide's about $60. Examine website, they do have fecal egg counting kits, and those range from $289 to $439. And those come with just about everything that you'll need, including a microscope to do a fecal egg count. So if you're interested, you could look at those. What else do we need to conduct a fecal egg count? We also need a microscope. And you could use a monocular or binocular scope. For me, I prefer to use a binocular. It's just more comfortable. You'd need at least a 10x eyepiece. It has to have some sort of light source. An internal light source is best, and a mechanical stage, even though not necessary. It makes it so much easier to move that slide so you can count those parasite eggs. And of course, some of these features will add cost to the microscope that you purchase. Additionally, you'll need flotation solution. And this could be simply a salt or sugar and salt. You can even use Epsom salt solution, salts to make this solution. Essentially, you're just trying to make a solution that's super saturated so that the eggs will float. There are commercial flotation solutions available out there, but I find it's cheaper to make your own flotation solution at home. There is a recipe over right beneath the microscopes, and we usually recommend pickling salt as opposed to using just regular table salt, because I believe it has anti-caking agents that doesn't dissolve pretty well. So if you're making your own, use pickling salt. Additionally, we found in our lab that these flotation solutions, they last for a pretty long time if they're kept in a good environment. So you want to keep them inside somewhere where there's not any fluctuation in temperature, and don't leave them out in your barn somewhere. I prefer to do the salt solution as opposed to sugar, just because with the sugar, it can tend to get moldy around the lid or on top of it, and it's kind of sticky and hard to work with. So I prefer to work with the salt solution as well. What else do you need? So for doing your MatMaster technique, you're going to need gloves for sampling. There's no risk of contracting parasites from these small ruminants. However, fecal matter, all fecal matter has bacteria. And so you do want to take care when you're collecting a fecal sample. Use a form of lubricant, especially on those first time animals that have never been sampled before. And regardless, you want to use some form of lubricant. If you don't have any lubricant, even your saliva works good. A small snap bag would be ideal for sampling, but not needed. If you have your glove, you could just collect the fecal sample in the glove, and just when you rip that glove off, you can leave the fecal sample in there and just take a Sharpie and label your glove. So you need a Sharpie to label. You'll need about 2 to 4 grams of fecal matter. 2 grams of fecal matter is approximately about eight fecal pellets. Um, Remember that if you have loose stools or excessive moisture in the stool, then that's going to make that sample heavier, just because it's moist. And so if it has a little bit of moisture, you will need less sample than if it's a dry sample. You'll also need a small cooler with ice. You would, if you're in the field and you know it's going to take you a while to get back to your home or lab or whatever, you're going to need to put that sample on ice, because if you leave it out too long in the heat and the humidity, then the eggs can hatch. At the same time, you don't want to freeze, you don't want to put these samples directly on ice or in a freezer, because they could freeze and damage the egg, and then they won't be able to float in the solution. You'll need a scale, but as I'll discuss when we start going through the steps, this is one that you could get or not. You'd also need some small cups to make your fecal slurry in, some tongue depressors, because you're going to need to break up that fecal pellet. You can also use craft sticks or spoons, they work good. A tea strainer or cheesecloth to get rid of the debris so you can count your eggs better, and a tuberculin syringe or pipette so you can fill your MatMaster slide with solution. So we're going to go in the next couple of slides, we're going to go over the steps of doing a modified MatMaster technique, and then we're going to go into looking at a video going over the steps again. So that will help you to see a better visual. But step 1 would include collecting and storing the fecal sample. So as I mentioned before, you'd want to use a rubber glove and lubrication to extract feces directly from the rectum. You can collect it off the soil or bedding or wherever if it is fresh, and you want to collect it from the top or center of that manure pack, so you don't get a lot of debris or dirt contaminating that sample. As I stated before, you need at least 2 grams of feces, and that's approximately 8 pellets. Again, if it's a really moist sample, then you need less. If it's a dry sample, then you need more. And the reason you can do anywhere from 2 to 4 grams. The more fecal sample you have, it does help with accuracy. So fecal sample storage, you're going to want to place the sample in a labeled snap bag or your gloves, as I mentioned before. Take all air, remove the air, seal it. If you're going immediately back to the lab to do fecal egg counts, that would work best. Fresh samples work best. If not, then you're going to want to keep the sample cool or keep it in a refrigerator until analysis. As I mentioned before, the eggs will hatch if they if they're abused by temperature and humidity. So if you leave them out for any extended period, then the eggs will hatch. And even if they've only been out for a number of hours, then you'll see when you're doing the counts, you can see little larvae moving around inside the eggs when you look at it in a microscope. It is ideal to do your analysis within a week of collection. Step 2, you would weigh out the amount of feces that you need to conduct your fecal egg count. So you weigh it out in a labeled cup, and I usually have just 2 cups stacked, one on top of the other, and you'll see this when I show the video later. And the outside cup is labeled, and I'll put both cups on the scale, I'll tare it, and then I'll add the fecal, I'll add the manure to it till I get anywhere from 2 to 4 grams. As I mentioned before, more increases accuracy. You also want to make note of the consistency, presence of blood, mucus, color, maybe if you see any tapeworm segments in that sample. If you do not have a scale, an easy way to actually estimate the weight of the fecal matter is to add 28 mils of saturated solution to a 50 ml centrifuge tube, and then add feces until that a volume of 30 milliliters is reached. So 1 ml is approximately 1 cc, so you could also use a 30 cc syringe to do this. So you could pull up 30 cc's of solution, and then you could then plunge that solution into a cup, and then you take off 2 cc's of solution, and that would be 28 if you want to figure out a way to get that 28 mils. But you can use a graduated cylinder, or you could use a dosing syringe to get your 28 or your 28 mils of solution. But again, you just want to make sure that once you have that 30 mil, if you're doing it in a cup or anything, you want to mark off where that 30 mil is. So if you're doing if you're using it in a cup and you pull up that 30 mils or 30 cc in a dosing syringe, you put it in a cup and just mark on that cup where that 30 cc is. Then you pull that 2 cc off and you add fecal matter until it's to that line. I hope that helps and makes more sense if you don't have a scale. Step 3 would be adding the saturated solution to fecal matter. So you'd mix those two together, the fecal matter or the fecal matter and the saturated solution, mix them together, and this is best achieved by using a craft stick or a tongue depressor, and you can also use the spoon, but you want to mix that out as good as possible. You want it to be a consistent, nice slurry. If you are using, depending on how much grams of fecal matter you're using, that will depend how much flotation solution that you're using. So the magic number, as I mentioned before, is 30 mils or 30 cc's. So if you use 2 grams of fecal matter, then you'd add 28 grams or 28 mils of flotation solution. If you're using 4 grams of fecal matter, then you add 26 mils. Both of them should total 30 mils. That's the magic number. And then you just mix them out. I really like these disposable cups to do fecal egg counts because they're sort of graded or rough on the inside, so I can actually mash that fecal sample. I could break it up easy on the inside of that cup. But some things that you could do if you have really hard or tough pellets are that when you collect the sample in a snap bag or collect it in a glove, you could take your hands and break up that fecal matter. Additionally, you could use a bottle or any other container and just gently massage that sample while it's in the bag or glove, just to help break it up a bit. And you can also leave that fecal sample to sit in the fecal solution for a couple of minutes so that it soaks up some of that moisture and becomes softer and easier to break up. So again, it's based on the principle of differential density. The parasite eggs are going to sink in water. They float in these dense solutions. The next step is to strain your fecal slurry into a clean labeled cup. So at this point, I'll usually pick the top cup off, and I'd use a tea strainer or a cheesecloth and strain that slurry into a separate cup. All right, so this helps to get rid of the debris, any debris or anything or large debris, including feed material, grass, all that stuff that might be in there, just to make it easier to view under a microscope. I prefer to use a double layered cheesecloth because it strains a lot better, and it gets rid of more debris and makes counting a lot easier. Step number 5 is transferring the slurry to the slide. So immediately after straining, you want to stir and fill your pipette or your syringe, and you could also use an eyedropper with a sample of the suspension, and then use that to fill both chambers of the MatMaster slide. Um, if you are used to doing this, then you can fill both chambers with one sample of the suspension. However, if you're still learning, because of the amount of time that you'll take to fill a chamber, you might want to fill one chamber, then stir the cup some more, stir the sample or suspension some more, take up another sample, and then fill the second chamber. And the reason why we say immediately after straining is because, remember, the eggs float. So if you leave this suspension anytime after you've strained it, if you leave it around sitting on the table, the eggs are going to float to the top. If you go in with your pipette or eyedropper or syringe, and you will tend to either go to the top or to the center of that solution, then that won't be a good estimate of the number of fecals that are dispersed in that suspension, because they're all at the top. So either you're going to get a really concentrated sample, or you're going to get one that's really diluted. So you really want to make sure that you stir that slurry before you pick up that sample, so that you're getting a good representation of those eggs that are dispersed in that sample. All right, and then you're going to want to let this sit for 2 to 5 minutes. And when I learned this technique, I was told not to let it sit for longer than an hour, but there are some labs that will say don't let it sit for more than 30 minutes, because that solution will start to evaporate out, and it will affect your counting. So 30 minutes might be a better way to go. Step 6, you're going to count the eggs that are inside the grid area. So no eggs that are outside the grid are going to be counted, only those that are inside the grid area. So using your 10x objective, you're going to count the number of eggs or oval shaped strong gel, sugar strong gel eggs that are in the sample, going up and down. Each chamber will have 6 lanes inscribed on it, and so you're going to want to count all these eggs that are in these lanes. You're not going to count strongyloides. Strongyloides are just, they're really small, or they're smaller oval-shaped eggs. Tapeworms or coccidia, you can make a note of the presence of these. And you're not going to count, as I mentioned before, any eggs that are outside of the chambers. So it's the reason why you're not going to count any tapeworm or coccidia, or whipworm or threadworm, or any that we're going to see on when we look at the diagram with parasites of ruminants, is because it's very unusual to see clinical problems when it comes to these worms. And the strongyloides, it's really more of a problem in it can be a problem in adults, but it's more of a problem in young animals. More older animals seem to be a bit more resistant to those. So this is a diagram showing you what the MatMaster slide looks like, and you'll see that when I do it in our lab and when I teach my students, I'll tell them to start at an edge. Start counting at the edge, not at a center edge, but one of the outside edge of that slide. And I can start, you can see the arrows showing you. So I would go count all the eggs within that lane, going all the way up Then I move across, I come down, move across, go up until I'm finished with that chamber, and then I move across to the other chamber. And then once you're done with that chamber and you move across, it'll come to the end of the slide. So even if you're too into counting, then you'll know that you've counted both chambers and not question, did I count both chambers or not? So that helps you to ensure that you're counting both chambers, and that you don't have to start over because you forgot where exactly you were. So what do you count when you're looking in those lanes or looking at that MatMaster slide? You're going to want to count all the barbapole and related parasites. And I know I've said this before, but those are your strong gel or strong gel eggs. They're oval in shape. You look at them in that diagram on there, they've got a presence of a shell, and then they've got a number of cells inside, and these are the cells that develop and form that larvae. I've mentioned also before that if there was any temperature abuse of that sample, then you'd actually see a developing larvae inside. You can actually make out the larvae, and you might see it moving around. So that's what you want to count. So this diagram depicts internal parasites of ruminants, and you'll see I've put stars above what you typically count when counting counting eggs using this technique. You'd count the Astertasia, Cuperia, Himancus, Strongylus, Aesophagastimum. So these are the ones that you would count when counting eggs using this technique. The others, as I mentioned before, other worms like tapeworms and the Nematodyrus or threadneck worm, all those you can make note of them separately if you think if you're seeing a lot in your samples, you can make notes of those separately. And the whipworm as well. The whipworm and the Nematodyrus, you can see they're, as well as the tapeworm, they're very distinctive. The Nematodyrus, there's no doubt when you've seen it, it is huge. And the Trichuris, it looks like a football. And the tapeworm actually has a triangular shape that you can see. We don't count coccidia eggs either when we're doing a fecal egg count. You can count them separately as well, but not in this total count that we're looking at. Again, we're looking at the larger oval-shaped worms, including your Himancus and your Trichostrongylus worms. Just some things that you'll see commonly when looking under the microscope at these slides. If you look in at that first image, you'll see a black circle with a clear center, and that's usually an air bubble. A few of these in your sample, it's no issue at all, no problem. You could still do your fecal egg count with a few of these. If when you're looking at your field of vision and it's filled with these air bubbles, then that's when you want to go back and take another sample for counting. Remember I said to stir your sample before filling up your MatMaster slide. You don't want to stir too hard because you can get a lot of air bubbles right in there. And sometimes you'll see pollen in there, you'll see other plant material in there. That sometimes might confuse you, and you might be wondering, should I count these or not? And you remember, you don't want to count anything but those strong gel eggs. Tapeworm, you can see a picture depicted there of what that looks like under the microscope. Again, you do not include it in your fecal egg count. You do not do that. You can make a note of it separately if you see a lot in your sample. The last diagram shows you coccidia, strongylid, and strongyloides. And you could see that the strongyloides are also oval shaped. They're just smaller than your strong gel eggs. And the strong gel eggs are what you want to count. And there are also some smaller coccidia eggs that are in that sample as well. But again, we're focusing on those strong gel or strong gel eggs. Step 7 then is to calculate the eggs per gram. So after you've counted both chambers, and it can be, when you're doing this without a counter, especially if there are a lot of eggs in there, it can be hard to keep track because you have to to basically, you have to be writing down or keeping track in your head some way how many eggs that you've counted. But I find a good tool is just a simply clicker, a simple clicker that you could use, and you click every time you see an egg, and that will help you to keep track of the number of eggs. But once you have your total number of eggs, so you're going to add the eggs that you have in both chambers, you're going to add them together, and then you're going to multiply these by a factor depending on how much fecal matter you used initially. So if you used 2 grams of fecal matter or manure, then you're going to multiply this number by 50. If you use 4 grams of fecal matter, then you're going to multiply this by 25. There is a calculation for how we come up with this factor. And if you're interested in getting that calculation, then you can email me. My email address was on the first slide, and I will send that to you. An easier way to remember is just to recall that it's 100 divided by the number of grams of fecal matter that you use. So 100 divided by 2 would be 50, 100 divided by 4 would be 25. And there's an example at the bottom of the screen that shows you how to calculate your total eggs per gram. And next, what I want to do is I'm going to show you the video that I prepared, and I'll just talk through the process again to show you what I was doing. So give me 1 second while I switch screens for you. And I hope you're able now to see the video. Okay, so there I am. I have my labeled bag. I have my lube in my pocket, and my colleague, Dr. Stefan Willius, is assisting by restraining the animal for me. So you'll need someone or some method of restraining the animal to take a fecal sample. No animal likes giving a fecal sample, so you'll need some way of doing that. As I mentioned, you'd need lubrication as well. If you don't have lube, then you will want to maybe use your saliva. If you try to take the sample and there's no fecal matter, sometimes if you use 2 fingers, you could spread them lightly, and that will stimulate the animal to defecate. Next, I am weighing out my sample. You can see my scale is tiered, and this sample is not, it's not like tiny pellets and dry. It has some moisture in it. So what I put out was pretty heavy. So I'm going to remove enough until it's about 4 grams fecal matter, so I can then add my solution. So I have my 4 grams of fecal matter, and now I'm going to add 26 mils of my salt solution. And again, remember, 4 grams of feces, the magic number is 30 mils. So if we estimate 1 gram of fecal matter is 1 mil, then 4 grams would be 4 mils. And so I only have to add 26 mils of solution. I'm using the tongue depressor to really break up that fecal pellet. Remember, those parasite eggs are inside that pellet, and I need to break that up to get the pellet out. Next, I'm going to be straining my fecal slurry, and this is to get rid of any plant material, any debris that might be in a sample. I'm going to show you 2 methods. I'm going to show you the strainer, the tea strainer, and I'm also going to do the cheesecloth. So you want to ensure that you've done a very good job breaking up that fecal sample. your solution or suspension should look similar, similar to what you're seeing here in the video. And then just take that tongue depressor and just try to get as much fluid as possible out. So the eggs are going to pass through that sieve, the strainer, whereas all the debris and other stuff that we don't want to look at under the microscope, these will be strained out. Now I'm using the cheesecloth. So all you'll do is just take a small square, put it over the mouth of the cup, make a small depression so you could add your suspension. And sometimes to get all the material, you want to stir that cup a bit so that you can get everything in that second cup. The next one I'm onto the cheesecloth. Next, I'm going to fold that cheesecloth onto itself and use my tongue depressor or craft stick to try to get as much solution out as possible. So there we have it, the solution. And now I'm ready to take some of that take a sample of that suspension and add to the chambers of the MatMaster slide. So again, you want to stir that sample not too vigorously because you don't want to have too many air bubbles in there, but you just want those eggs to be distributed in that sample pretty good so you get a good representation of it. So I take one sample, I fill one chamber. Look at me spilling right there, too aggressive. Then I take a second sample and fill that second chamber. So this does take some practice, and the easiest way to do it is to do it like how you see me see it done on this video, is to start at one corner and try to make sure there's not a bubble at the tip of an air bubble at the tip of that syringe, because that will introduce an air bubble to that chamber, and they can be difficult to get rid of at times. So try to make sure that you squeeze it and get rid of any air bubble that might be at that tip. So next you're going to let that MatMaster slide sit for 2 to 5 minutes, ideally 5 minutes, so that those eggs could float to the top. Now what you're seeing right now is what you'll see when you put it under the 10x magnification of a scope, right? This is what you'll see. So you'll see right between those two lanes, and you'll count all the eggs, all those strong gel eggs that you'll see. This was a bad sample. I will tell you, we've got here sheep at Virginia State University, and it's pretty good news for us in that this time of year, there were no eggs that I could find in these samples. Bad for you that you can't see any eggs, but I've got another slide that I'll show in a couple of minutes that will show you some eggs. So you can see there's some small amount of air bubbles that are in there. That's not bad. That's fine. You can still count with those air bubbles. If you saw it widely distributed across the screen, this is where you would want to clean, to remove that solution from under the chamber, clean it and refill to look at a new sample. But you can see there's nothing. So I'm just simply going up a lane right now. I'm going to come across and go down. And once I've finished all 6, then I will move to the other chamber and count the 6 lanes on that side. I'm going to hurry this up a bit because I did not see any eggs on this slide. So I'm going to move forward to another slide where I had some eggs. All right, so here's another slide, and the view on this might not be so good, but that air bubble that's in the center of the screen, you'll see that there are 2 eggs on that view, 3 eggs now as we're moving down. So I've spotted 3 eggs so far going down that lane. Here's a 4th one right there in the center going down. And those I would count, make note of. And this time I've started from the opposite edge of the slide. And so I'm going right as opposed to left in the other slide. So as you're going through these slides, you might want to refocus using the fine focus so you could see things. As you move up and down, you might lose focus, and you might want to play with that fine focus some so that you're able to see clearly. But essentially, this is basically what you do. You go up and down each lane and count the total number of eggs. There's another egg again in the bottom left corner. There's a tapeworm egg that we're going by right now. And there's another tapeworm on the left corner again. Again, we count everything that's inside the grid and nothing that is outside the grid. So I would continue this slide until I've counted all the eggs that are present. All right, I'm going to move further along in this video. Okay, and this is just to show you. I tried to zoom in a little bit more to show you some eggs. That's air bubbles right there, and I'm using my fine focus to focus in and out, as you can see. This isn't such a good view, but I hope you're able to make out those air bubbles. Right now, above the air bubble, you'll see a strong gel egg, so it's above, and I'm trying to focus in and out so you could see it. You'll see that other shell, and then the cells contained in the middle. And so that's about the size that you're looking to count. I'm moving it across so we could see, to see what else we can see under this slide, and I think I was able to pick up a tapeworm. So right beneath where you see that tapeworm, there was a tapeworm right on that slide. So again, we're looking to count only the strong gel and trichos strong gel eggs that are in that view. Okay, so I've stopped share, and let me now go back to my presentation. Okay, all right, so once we finish counting all those eggs, as I said before, then we'll add those eggs together, and then we'll multiply by that factor that we mentioned before. Again, 2 grams of fecal matter, it's 50. You multiply the total eggs by 50. If you use 4 grams of fecal matter or manure, then you'll multiply that by 25. And that sample, that even the second sample I looked at, the first sample had 0 eggs. The second sample had a few eggs, and even then, I think total, when I totaled it, it was less than 20 eggs, and that would have been total probably less than 1,000 eggs per gram. So some limitations of fecal egg counts that we should keep in mind when we're doing this technique on our farms or in our labs. Fecal egg counts, at times they're not very accurate, especially when they're low. So the lower that fecal egg count or the lower the level of infestation in your herd, then it's not very accurate. Worms do vary in their egg producing capacity, so that will affect the fecal egg count as well. Even if you take multiple fecal egg counts throughout the day, that the fecal eggs aren't distributed uniformly throughout that fecal sample, so they could vary in a sample as well. Inhibited larvae do not lay eggs, so if they're in their hypobiotic stage inside the animals, they aren't laying eggs. So even though they're inside the animals, they're not laying eggs, so it will not indicate whether they have the presence of worms inside or not. Strong gel eggs, they all look the same, and they cannot be differentiated at the egg state. In order to do this, you'd have to use other techniques, such as a larval identification or peanut agglutination test to identify homuncus from the other parasites. I mentioned before, eggs are not distributed evenly in a fecal sample. Loose stools or diarrhea underestimate the fecal egg count. That moisture increases the weight of that fecal that fecal sample, so it underestimates that fecal egg count. And finally, procedures will vary by labs and technicians. So the procedure I've showed you today is what I do in my lab. If you watch someone else, or you go to someone else's lab class or lab to learn, then that technique might be slightly different. The key thing to remember is that you want to be consistent in how you're doing it on your farm or in your lab. So consistency is important. The final thing I want to point out is the website for the American Consortium for Small Ruminant Parasite Control, wormx.info. This is the go-to site for all things related to small ruminant parasites. It's a group of scientists, veterinarians, extension specialists who are devoted to studying and developing new methods for sustainable control of internal parasites in small ruminants. So this completes my presentation, and I hope, I wish you good luck in doing this technique in your labs or on your farms. And if you have any questions, feel free to reach out to me. Thanks.
Thank you, Dr. O'Brien, for giving a very detailed and outstanding presentation on how to conduct fecal egg counts and what to do with that information. As I mentioned before, this is one of 8 webinars in our weekly worm webinar series. Be sure to tune in to some of the other ones or watch the recordings. Again, this webinar is sponsored by University of Maryland, Delaware State University, Virginia State University, and Fort Valley State University. For any questions about parasites, as Dr. O'Brien mentioned, be sure to visit the website of the American Consortium for Small Ruminant Parasite Control, wormx.info, or feel free to contact any of us with your questions about parasites.
All right, thank you.